Okay, we're back for part two, and now we're going to start looking at some more subtle interactions within this pathway. Specifically, starting to order the pathway and really demonstrate that PI3 kinase is upstream of AKT in the phospho-Bad pathway, and also try to link the pathway to cell survival. We haven't really seen that yet. Well, let's dive in and see what we see. So, to begin, we're going to switch gears a little bit and we're going to look at a slightly different readout of bad phosphorylation. And what I mean by readout is simply a measurement. And right now, the readouts that we've been using have been looking at, bands on a gel to demonstrate phosphorylation of bad. And that's because the bands are there because of the transfer of radioactive phosphate to bad. So we can measure that transfer as a band intensity. Now we're going to start looking at something called a mobility shift. A mobility shift, as the name implies, is a change in the way a protein moves on a gel, a shift in its mobility. And so let's keep this band here. Let's imagine that this is bad in the unphosphorylated state. When we add a phosphate, we've added weight. So when we run this on a gel, phosphobad runs a little bit higher in the gel than regular bad. So if our band intensity that I'm outlining here represents unphosphorylated bad, this mobility shift up here represents phosphorylated bad. So the presence of that phosphate makes bad run a little bit slower on the gel, and we get the mobility shift, okay? It's another way, another readout that indicates phosphorylation. So let's take a look at the mobility shift in vivo. And note that these are biochemists, and so in vivo to them means in intact cells, not in the animal per se, as we typically think of in vivo meaning. So now what we're doing is we have cultured cells. We stimulate with the growth factor. So anytime we see a plus here, we know that we've stimulated with the growth factor. The ligand binds to the receptor, it sets off the signaling cascade. And now we take bad out of those cells, out of those cell lysates, and we run it on a gel. And we ask, do we see the mobility shift or not? So notice how they're drawing this with two arrows, much like I've indicated here. The top arrow is the mobility shift. In the absence of the growth factor, we do not have the mobility shift. So take a look at this lane of the gel. You don't see that subtle little top band. But when you stimulate with the growth factor, we now see the mobility shift in this lane of the gel. And I'm not going to try to circle that mobility shift because it will obliterate the subtle shift in weight. Adding the phosphate is just adding a little bit of extra weight. So it's not a huge change, but it's a change nonetheless. So now we can start using inhibitors. Remember that inhibitors are part of our arsenal of tools, and the technique of using an inhibitor to dissect a signaling pathway is very common. So remember earlier they used this inhibitor called WORT and LY. These are both PI3 kinase inhibitors. And in their previous figure, I believe figure 1, they showed us that the in vitro kinase assay was impacted by the use of the PI3 kinase inhibitor, and that suggested that PI3K was in the pathway that leads to bad phosphorylation. Now they're going to show us the same thing, this redundant result, but now in vivo. So now they treat with the inhibitor, either the Wortmannin or the Wortmannin. Let's just look at this first. They treat with the PI3 kinase inhibitor, they stimulate with the growth factor, and take a look, guys, we've lost the mobility shift. So that tells us that, growth factor and PI3 kinase are in the pathway that leads to bad phosphorylation. Same thing that they've seen before, now just showing it in intact cells. And as we would expect them to do, they tried a panel of other inhibitors, and they've shown data for some of them. PD, the one that I mistakenly circled before, that's a protein kinase C inhibitor. And you see that inhibiting protein kinase C does not change the mobility shift. So we know that PKC is not in the pathway that leads to bad phosphorylation. Similarly, pretreating with the P70S6K inhibitor called rapamycin, that one doesn't change the mobility shift either, so we know that that kinase is also not in the pathway. The only one that eliminates the mobility shift is wortmannin, and we see that again here. Okay, so another way of looking at bad phosphorylation and another way of placing PI3 kinase in the pathway. Now, let's see if we can demonstrate the order of the pathway and specifically try to show that, this arrow in the pathway is AKT and it's downstream of PI3 kinase. So the way that we do this is we use an inhibitor of PI3 kinase. So we can use wortmannin or LY to inhibit PI3 kinase, show that bad phosphorylation goes away, so we lose the mobility shift. But then if we put in overactive AKT that doesn't need the upstream pathway to become turned on, if we can restore bad phosphorylation even in the presence of the PI3 kinase inhibitor, then we know that AKT is downstream of PI3 kinase in the pathway. So the way we run this experiment is we have our cells that we're culturing, we treat them with the inhibitors or not, and then we treat with the growth factor ligand or not. We make a lysate, we pull bad out, and then we look for the mobility shift. And let's take a look at what we see. And first we're going to focus on these data down here. And just remember, here, instead of transfecting in an active version of AKT, they're just putting in an empty vector. So no active AKT. And what we're looking at here, the band on the gel, is actually bad. Okay? And I hope you can see that in this lane, there's the mobility shift. So let's take a look. So treat with no growth factor. That means we don't stimulate the pathway at all. And of course, we don't see the mobility shift in that lane. No surprise there. Treat with the growth factor. Don't add the PI3 kinase inhibitor. Get the mobility shift. Great. So they've replicated what we saw in the upper panel on this figure. Now let's do that again in the presence of the inhibitor. Now we add the PI3 kinase inhibitor, and either one of them eliminates the mobility shift in those two lanes. So all that looks exactly like we had seen in this figure. They've essentially replicated it once again. What's different about the top part of this gel in Western blot is now, in addition to stimulating with the growth factor, adding the inhibitors, they're also transfecting in overactive AKT that's always on. Okay? And now, after we stimulate with the growth factor, we get the mobility shift as we saw before, and also in the presence of the inhibitor. You can see that extra band up top there, and that represents activated, or rather phosphorylated bad. So the bottom line here is that active AKT that's always on and doesn't need the upstream members of the signaling pathway can override the PI3 kinase inhibitor, and that places AKT downstream of PI3K in the pathway. Now that we've established this relationship that PI3 kinase is upstream of AKT in the growth factor pathway that leads to bad phosphorylation, we're going to further explore this relationship. And we're going to see whether or not phosphorylation of bad occurs at position 136 downstream of AKT in the pathway. So they're zeroing in on the site of phosphorylation, as they've done before, and really convincing us that AKT is what's phosphorylating bad at that position. So now they do a co-transfection of their cells, both with AKT of various versions, either inactive or active. And then they also transfect bad at the same time in either wild type bad or the S136 mutant bad. And now they ask, does phosphorylation of bad occur? They go back to using their phosphorylation specific antibody. And when we co-transfect bad along with active AKT, we get a nice thick band that we see there on the gel that indicates phosphorylation has occurred. Don't worry about these other faint bands that we're seeing here. These are background bands. We don't have to consider those. What we're looking at is the real intense expression that we see right there. Notice what happens when we transfect in inactive AKT. We lose that nice thick band. So that really helps us to understand that it's active AKT that's required to phosphorylate bad. And I know I just circled this, but they show us that that real intense band that we see here is lost when we deliver the S136A mutant version of bad. If we take away that phosphorylation site at position 136, we lose AKT phosphorylation of bad. And so that tells us again that not only is AKT acting on bad as a kinase to phosphorylate it, it's doing so at position 136. And the pattern of data is the same for the double mutant. And if you're wondering what the bad Western blot is down here, this isn't phosphobad, it's just looking at total bad. Okay, and unphosphorylated bad is what this is measuring. And it's showing us that we successfully delivered bad via transfection every time we said we did. Okay, if AKT is truly the kinase that phosphorylates bad, then we ought to see these two things interacting. And the way we look at protein-protein interaction is via co-immunoprecipitation. So what they do here is they have their cell lysate, and it contains bad and AKT, and they've done it under conditions that would preserve the physical interaction. So in their tube where their cell lysate is, this interaction between bad and AKT is occurring. They use an antibody raised against bad to pull this complex out. And then they run a gel where they can, separate out bad and AKT. If they can go in and Western blot AKT on this gel, then we know that the only reason and the only way that AKT got there is because it co-precipitated with bad. Using the bad antibody pulls out AKT indirectly because of their interaction. So they transfect bad along with their various versions of AKT, and then they co-precipitate using the bad antibody, they IP with bad, and they Western blot with what they're calling anti-HA, but that's just a way of detecting AKT in this experiment. And you can take a look at the AKT band. We see it here in these three lanes where we know we've delivered AKT. So when we have bad and AKT both present in the lysate, we can pull out AKT by using a bad antibody. In other words, they co-precipitate, that means they interact. And if you're wondering what the rest of this stuff is, they just wanted to show us with this Western blot down here that when they told us they transfected AKT in these three lanes, indeed we see it in those three lanes. And yeah, there's a whole bunch of other background stuff that goes on here. This is very common in Western blots because of cross-reactivity with antibodies. But the specific one that runs at the right molecular weight is the one that I circled. And we only see that when we deliver AKT. Okay, so AKT and bad physically interact. And that interaction doesn't depend upon the phosphorylation state of bad. This is a kind of follow-up experiment that they do to show us that, phosphorylation at position 136 does not impact the interaction. And that's not surprising. We wouldn't expect it to impact the interaction. It's just another way of driving home the point that AKT is the kinase that phosphorylates bad at position 136. So same kind of experimental design as before. We co-transfect our AKT with our different versions of bad, and then we use bad antibody to immunoprecipitate, and then we can Western blot AKT as we show here in the presence of bad. So the only way AKT got there is by that physical interaction, and notice that AKT interacts with the unphosphorylated version of bad as well. So the version of bad that cannot accept a phosphate at position 136 still interacts with AKT. And that's just another way of really driving home the point that AKT is immediately upstream of bad in this pathway, and this is the kinase that does this phosphorylation. All right. So far, we've seen a lot of biochemistry, and we've seen a lot of things like kinase assays, co-immunoprecipitations to look at interactions. What we haven't yet seen is the real good demonstration that the result of this pathway that produces bad phosphorylation at serine position 136 leads to cell survival. Now they're finally going to link growth factors and bad phosphorylation state to cell survival in the pathway to really drive home the point. So how do they do this? Well, they have a way of measuring in their cell cultures the percent of apoptotic cells. Okay? And they can manipulate a whole bunch of different variables. For example, they can transfect into these cells various versions of bad. You've got your wild type version, your 112 mutant, your 136 mutant, and your double mutant. Okay, so that's what they do. Then for half the cultures, they continue to treat with the growth factor that gets this whole cascade started. And the other ones, they remove the growth factor from the culture media. They call that starvation. And we would expect starvation to elevate apoptosis, and of course it does. So let's take a look at the pattern of data that we see here. So transfecting in wild type bad and adding the growth factor means that we can stimulate the pathway because we're adding the ligand that gets the whole thing started, and it'll lead to some bad phosphorylation, which will provide some cell survival. So we're not at 100% apoptosis or something like that. We're down to about 40% apoptosis. And we're basically at the same level when we deliver the 112 mutant. Remember, the 112 mutant still contains the serine at position 136. So phosphorylation in the pathway can still occur, and that'll lead to more cell survival, which it does. Okay. What happens if instead of putting in the 112 mutant construct, we put in the 136 alanine mutant construct? Now we've eliminated the phosphorylation site, so that can't happen. We should boost apoptosis if our hypothesis is supported. And indeed we do. So look at that. We're now up to 80% apoptosis, even though we're stimulating with the growth factor. And that's because we've delivered extra bad that can't be phosphorylated and can't cause cell survival. And the double mutant does the exact same thing. Really cool. Over here, it's basically the same pattern of data, but everything's amplified. And even for the wild type and the 112 mutant, we elevate apoptosis because we're not adding the growth factor that gets this whole thing started. No real surprise there either. So they've successfully been able to show that activation of bad phosphorylation at position 136 enhances cell survival. Now, we're going to take that idea and we're going to extend it to show that AKT is involved in the pathway. So let's take a look. Let's draw the pathway again one more time and make sure that we follow the logic here. It's really kind of cool. So that whole pathway is going to lead to cell survival. Okay, so experimental design, similar to what we saw before. We've got two different treatments here. This one is with our culture media plus growth factor. I don't know why they didn't label it that way, but that's what it is if you look at the methods. So they add the growth factor. And down here, it's the same idea, but they starve. They take the growth factor away. Okay. This part of the figure is essentially a repeat of what we saw in the previous one, but let's walk through it anyway. So empty vector transfected, okay, with growth factor, very little apoptosis because we're stimulating the pathway, no surprise there. take away the growth factor through starvation and dramatically, boost the level from about 5% background to about 40%, again, just like we've seen before in the previous figure. Now we do that in the context of wild type bad, and wow, look what happens with no growth factor present. We're up to almost 100% of the cells apoptosing. Whereas here, when we add the growth factor, we keep it at around, under 40% like we'd seen before. Same pattern of data when position 136 is available for phosphorylation. If we take away that position, we're back up to what we saw before. And again, the whole pattern is sort of amplified by starvation, just as we saw before. So what's different? Why are they even showing these data again? Well, they want to compare it directly to what happens if you co-transfect constantly active AKT along with wild-type bad. Now here's where it gets really cool. Remember, if we put in active AKT, it can directly phosphorylate bad and lead to cell survival, even in the absence of any of the upstream components of the pathway. So now, even when we take the growth factor away through starvation, we suppress dramatically the percent of apoptosis in the culture. And that's because we don't need the growth factor when we co-transfect the active AKT. It's able to override the absence of the growth factor. And again, that places AKT pretty downstream in the pathway, just as we've seen before. And we know that the phosphorylation state of bad is what leads to cell survival because of this result right here. When the serine at position 136 is still available for phosphorylation, it doesn't matter if we take the growth factor away. As long as we deliver the active AKT, we still get the bad phosphorylation, still get the cell survival. Look what happens once we take that site away. We're back up to that 80+% apoptosis. Okay? And of course, active AKT can't override if we put in a version of bad that can't be phosphorylated. So now we don't get the enhanced cell survival. And the pattern of data is very much the same up here, just everything's a little bit lower because there's background growth factor to take care of any endogenous bad in that pathway. So here what the authors have done is they've linked the growth factor to cell survival, and they've shown that the phosphorylation state of bad is a critical regulator of the growth factor's ability to enhance cell survival. I hope you agree that they've really done an excellent job of dissecting this pathway. All right, let's do a quick summary. So what the authors have been able to do is show us that bad is able to be phosphorylated by AKT both in vitro and in vivo. So AKT is directly upstream of bad in this signaling pathway. The position that it occurs on is serine at 136. And it occurs in a PI3 kinase and growth factor dependent manner. And once we activate bad phosphorylation at position 136, we lead to cell survival and inhibit cell death. They've even really convinced us that AKT and bad are directly together in the pathway by showing us that they physically interact via the co-IP. So the authors have done a variety of different things in a variety of different systems to all point to the same conclusion. So this is an important paper in the cell death field because it was the first time we were able to say that AKT is definitively the kinase that phosphorylates bad. And that's important. It was important for us to understand the mechanism whereby bad phosphorylation is linked to cell survival. And they showed us that phosphorylation state is critically linked to the prevention of cell death. And the other things they were able to do, they were able to map the phosphorylation site and show it was serine at position 136 that was so critical, and show the physical interaction. Now look, no paper is perfect, and no paper can address every single question. There were parts of the mechanism that weren't figured out and that a few subsequent papers did. One of the things that they demonstrated later is that when bad is phosphorylated, the way that we prevent its interaction with BCL2 is to have it physically interact with a protein called 1433 instead. That has to be the most bizarre name of a protein ever, 1433. That is the name nonetheless. 1433 is a cytosolic protein that binds to phosphorylated BAD and sequesters it. So it traps it, and it keeps it from going over to the mitochondrion where it would physically interact with BCL2 and pull BCL2 away from Bax. And there's other things that they didn't do in this experiment that we could ding them on if we wanted to be really critical. Usually, we like to see co-IP with what we call endogenous levels of proteins as well as overexpressed. We want to make sure that the interaction isn't artificial. This work has since been done, and we do see the interaction between AKT and BAD, whether we overexpress or rely only on endogenous levels. Similarly, they didn't do some downstream aspects of the pathway, like, for example, they didn't show that what leads to cell survival or apoptosis is the regulation of the downstream caspases. Again, subsequent work has shown that when BAD is phosphorylated, caspase activation doesn't occur. And finally, there was this whole idea that they played up about protein kinase A phosphorylating BAD at position 112. You may remember that one. They didn't really follow up on that and tell us what the consequence of that was. It does not seem to be linked to the cell survival pathway, but what is it involved in? We didn't know. Nonetheless, the work is really quite well done. Okay, so implications and discussion, just a little bit here on this. Remember that understanding apoptosis is important for a variety of reasons. First of all, just from a basic science perspective, understanding how structures are sculpted during development, understanding how neurons projecting into a target, their number is matched to the size of the target. It helps us to understand all these important things. It also has clinical relevance because a very large number of human diseases result from dysregulated apoptosis. And almost all of the neurodegenerative diseases, Alzheimer's, Parkinson's, etc., have inappropriate apoptosis. And so understanding how to regulate it may be helpful therapeutically. Maybe we could think of a way of inhibiting this process, for example, and treat the disease in some speculative way. Also, many diseases suffer from inappropriate lack of apoptosis, and cancer is a good example of that. What if there were a way to tap into this endogenous apoptosis network to cause a cell that you don't want around to die? That could be very helpful therapeutically. And not surprisingly, many labs are looking at whether this pathway could be exploited clinically. Maybe we could figure out a way to inhibit apoptosis to treat degenerative disease, and maybe we can figure out a way to drive it to treat some forms of cancer. So figuring out how to regulate caspases is an important part of that question. All right, so what you've seen me do here is dissect this paper, and I hope you see that this paper really is a good microcosm of this third unit, where we've been looking at ligands, receptors, signaling pathways, and the tools and techniques to dissect these pathways. This paper really provides a nice way of encapsulating the third unit. So this is the end of the third unit, and we will have one more lecture on Wednesday. Dr. Shaven will be back, and she'll be talking to us about the use of stem cells, what they are, and is there a potential use for those therapeutically or clinically. And it'll be just kind of a fun little diversionary way of looking at a different kind of topic than the one that we've been focused on so much during this third unit. It's a very good lecture, so please give it a listen and enjoy. And I've got one more slide that I'm going to walk through here, and I just wanted to have some final comments on the course and give you sort of a little bit of closure on what we've done this term. So I'll do that in the next slide. Please listen to that as well. Okay, I wanted to spend just a few minutes here summarizing what we've done this term, and I've got a few other comments that I want to make as we sort of finish out. Obviously, this is the most unique term that any of us have experienced as professor or student. I want to thank you all for your attention, your professionalism, and for hanging in there during, obviously a difficult and not ideal set of circumstances. I'd like to thank my teaching partner, Dr. Shaven, and I appreciate her work and hard work during this term. And I really also want to extend a word of thanks to all the TAs that have made this class possible. Literally, the class could not have run without their hard work, and I thank each and every one of them. It has been a big challenge for them to be balancing their normal load of coursework and working with grading and answering questions, etc., during this term. And I'm very grateful to all of them for their help. So let's just quickly recap where we've been and what we've done. It's always important to look back and reflect on the growth, hopefully, that we've achieved during the term. I want to remind you that the course was really designed into thirds, and the first third was spent defining the cell and its study. So it's really looking at structure-function. But in addition to looking at structure-function relationships in the cell, we wanted to lay out an experimental framework because we wanted you to be able to understand how we know what we know. And by laying out some key model systems and experiments early on, we were able to then take an empirical approach to new material as we moved through. So after we defined the prototypical cell in Unit 1, we moved on to Unit 2 and we looked at information flow in a cell. And really what we were focused on here is the central dogma, DNA to RNA to protein, and trying to understand how this is regulated. How do we regulate the replication of DNA, the transcription of DNA into mRNA or RNA in general, the translation of mRNA into protein? And furthermore, how do proteins get modified post-translationally by things like proteolysis or phosphorylation, for example? So looking at how information in the form of sequence of nucleotides flows through the cell set the stage for the next unit where we looked at the proteins themselves. So once we were at the protein level, we now looked at those protein networks and how we could tap into those protein networks. So the ligands, the receptors, the signaling pathways, etc. And then overlaid onto that all of the experimental approaches that culminated in the paper that we've just discussed here in this lecture. So the other thing we need to remember is that interwoven into all of this was the laboratory portion of the course. And what we tried to do in the lab was to have a project-based look at a question in science. And the question was, what's the role of proteolysis in producing this amyloid beta fragment that leads to Alzheimer's disease, and how might we think about altering those dynamics? And so, of course, you'll recall that what we did was we upregulated alpha secretase activity and were able to show our hypothesis was supported. We got a downregulation of amyloid beta levels. And what I think is cool about the lab here, in addition to its sort of project-based design, is the fact that a lot of the techniques that we're utilizing in the lab are some that we saw throughout these units in the lecture. And to the extent possible, we have attempted to integrate the lab material and the lecture material together, to integrate them so that it seems a coherent whole. And we certainly all hope that that came across. Okay, so that was the course, and you are essentially done. We have one more lecture on Wednesday. All right, don't forget about the last set of discussion questions for lecture and lab that are due this week. And good luck. The last thing I'll leave you with is a reminder to stay safe, and hopefully we will all be together in fall. That's certainly my wish. And I want to say again, thank you, students, and good luck to you. And please don't hesitate to stop by and chat anytime about your experience in the course, questions you had related to the course or the lab, or life in general. It's always something I enjoy doing. So take care and hope to see you soon. Bye-bye.